Welcome to this episode of Substantial Matters, Life and Science of Parkinson's. I'm your host, Dan Keller. At the Parkinson's Foundation, we want all people with Parkinson's and their families to get the care and support they need. Better care starts with better research and leads to better lives. In this podcast series, we highlight the fruits of that research, the treatments and techniques that can help you live a better life now, as well as research that can bring a better tomorrow. For many years, levodopa has been the treatment of choice for Parkinson's because of its effect in reducing motor symptoms. But the disease still progresses regardless of treatment, and attempts to protect neurons in the brain and slow the progression of Parkinson's have not been successful. Advances in modern genetics now open a new window on possible ways to look at the underlying causes of the disease. They can also help select people for clinical trials who have the most likelihood of benefiting from a particular experimental treatment. Today, Dr. David Simon, director of the Movement Disorders Center at Beth Israel Deaconess Medical Center in Boston, a Parkinson's Foundation Center of Excellence, explains how genetics can be a guide to developing new drugs that might preserve nerve cells and potentially keep Parkinson's from progressing. In our discussion, he mentions two terms that may not be familiar to everyone. One is biomarker, a shortened term for biological marker. Biomarkers are characteristics of the body that you can measure. They are important to medicine in general and to drug development in particular. Biomarkers tell us how the body is doing and can help identify disease risk or disease progression. The other term is alpha-synuclein, a protein found in the human brain that is associated with the development of Parkinson's, so it is a potential target for drug therapy. Dr. Simon sets the stage for the situation today and how scientists are moving the field ahead to try to find ways to slow the progression of Parkinson's.
It would really dramatically change the way we treat Parkinson's disease if we could find one or preferably more strategies that can slow progression.
So what are some strategies? How can we gear up to know better how to attack it?
So the first step, of course, is understanding what causes the disease to progress. In some cases, it's what causes people to get the disease in the first place will also be a mechanism relevant to why it progresses, although in other cases, causes of progression may be distinct from triggers of the disease. I think one of the important clues is genetics, where we can have a very clear cause association of a change in a gene with high risk of developing the disease, and that can give us a great clue into a target for neuroprotection. But there are other strategies as well for identifying promising mechanisms.
The genes that are being looked at now, are they associated 100% with Parkinson's disease, or are they just another one in the mix that raises the risk or modifies it?
Yeah, so the genetics of Parkinson's are very complex, and both types that you just named have been identified, although there's also still a lot about the genetics of Parkinson's we don't understand. In terms of a single gene in which a mutation is found that puts you at very high risk for getting the disease, it's only a very small percentage of patients in which we can identify such a mutation. There are common variants, sometimes in the same genes, that have one of those other types of mutations I mentioned, where a common variant has a small impact on your risk of the disease. And then there's some in between, where a variant puts you at high risk of the disease, but not everybody who gets that mutation will get the disease. And I can give examples of those if you wish.
Sure.
One would be a gene called the LRRK2 or LRRK2 mutation. Maybe 2% of all Parkinson's patients have this, which is actually relatively common as far as high-risk genetic mutation goes. It's a mutation that we're still working to try to understand exactly why it causes a high risk of Parkinson's, but it's probably due to increased activity of the enzyme produced by that gene. And that gives a great clue as to a possible neuroprotective strategy. In patients who have a mutation in this gene, we know that high levels of this enzyme cause them to either get the disease or at least have a high risk of getting the disease, and we can measure the activity of that enzyme. And so many pharmaceutical companies are designing drugs that specifically inhibit that enzyme. And it makes a lot of sense then that those drugs might help to slow the progression. Now, we can't know that until we test it properly in placebo-controlled trials, and it takes a lot of work to get a drug like that, even to the beginning stages of trials, but that work is being done. And I think that one reason that has greater promise than some of the past strategies is that it's targeting a very specific mechanism in a subset of patients who we can identify as being patients with the greatest chance of benefiting from the drug. So if we tested one of these LRRK2 kinase inhibitors in all Parkinson's patients unselected, we might not see any effect because it may be only that 2% with the mutations could benefit. But we have a way to select them. We can test patients and select only those with the mutation for the initial clinical trials. And, disappointingly, that means if it works, we've got a great strategy, but only for 2% of Parkinson's patients. On the other hand, with so many failures, with promising agents that have not shown efficacy in clinical trials, if we can make progress in a small percentage of patients, that would be a huge victory. And then we just do it again in another subset and again in another subset, and eventually, hopefully, we'll reach a large group of Parkinson's patients.
That gene would be a biomarker for who may best be in a trial or benefit. Are there biomarkers to follow the therapy itself, or do you have to wait years to see that something is working or not?
There are many different types of biomarkers. So you can have a biomarker of the drug effect, and you can measure that very quickly. So you can measure, as I mentioned, the activity of this enzyme and treat cells in a dish, or maybe even a person, and draw a blood sample and measure to see if the drug's having the desired effect. That's very quick. That's great. And that we can do for several drugs. And it's important because some drugs have gone to neuroprotection trials, in clinical trials that have failed, where we don't really know for sure what the mechanism is. So the drugs failed, but we don't really even know if the drug was having the necessary mechanistic effect in patients that we wanted, because we either can't measure it or we don't know what to measure. With LRRK2 kinase inhibitors, we can measure the activity of this enzyme. It's having that. We know it's having the desired effect. If it doesn't guarantee the trial will work, but if it doesn't, we've got good arguments why this mechanism isn't what we think it was. But with genetic data, I think we have strong reasons to think we're on the right track with that mechanism. So other biomarkers that would be hugely valuable would be a biomarker of progression. Thankfully, Parkinson's does progress relatively slowly in most patients. That's great for patients, but we want to make it progress even slower. To do that, we can't just treat patients for a month and see if they progress slower, because that's really not enough time to, detect progression. If we had a biomarker of progression that was more precise than our clinical exams, we might be able to detect that with a shorter amount of time. With some strategies, the biomarker could tell us we're on the right track even before we've had time to detect clinical progression. One example would be there are vaccine studies underway to clear away alpha-synuclein. There's an overwhelming body of evidence implicating alpha-synuclein in the degenerative process in Parkinson's. So it could be in a short period of time, there was ways to image alpha-synuclein in the brain, which, by the way, there's not yet, but a huge amount of work to develop that type of a biomarker. And if we had that, maybe we treat patients for the 1 month I mentioned and show that we've cleared away alpha-synuclein. And if we do that, then it's not exactly a biomarker of progression, but it's a biomarker that the drug is affecting a relevant pathophysiological pathway, and then you'd know that's worth pursuing in a big, longer phase, expensive trial. On the other hand, if you had a drug or a vaccine that you hoped would clear away alpha-synuclein and you don't see any evidence of that, maybe you stop that there and put your resources towards a more promising agent.
So where do we stand now in terms of prospects? I mean, we have all these tools in hand. Are these going to open up the field in a different way from what we've looked at in the past?
One way it's different is, as I mentioned, now we're targeting defined subgroups. So, a couple examples of trials that failed that didn't do that would be coenzyme Q10, which affects mitochondrial function, meaning energy metabolism, and maybe has antioxidant activities and some other mechanisms. But there weren't specific patients who we would think would be likely to benefit compared to others. The same thing for creatine, a drug with some overlapping mechanisms, also involving energy metabolism and antioxidant defenses. And both drugs took pretty much all Parkinson's patients. I mean, there were some entry criteria, but it wasn't specific to the mechanism of these drugs. And although they looked promising in preclinical data, they both showed no benefits in phase 3 clinical trials. I think a drug like an inhibitor of LRRK2 kinase, where you can identify specific subsets of patients who are likely to benefit have a much greater chance of success. Another example would be another gene, the glucocerebrosidase gene. This is a gene involving Gaucher's disease, a completely different disease, but it was noted that people with Gaucher's disease have a high risk of later developing Parkinson's. This was noted especially after treatment for Gaucher's disease got better and people were living longer, and this became clear. But people who are carriers of a Gaucher's mutation, they don't get Gaucher's symptoms, but it was noted they too are at higher risk of Parkinson's. And these mutations cause loss of the enzyme activity. Some companies are working on other drugs that increase the activity of that enzyme. I'm actually working with one company that's doing that, but there are others as well, including one that's moving into clinical trials right now. So there you could identify patients either by having a mutation in this gene where you know they have very low activity, or even there are some patients who don't have a mutation in the gene that you can identify, but still their activity is low, and you can enroll patients with this low enzyme activity and hope that they would have a greater chance than others at responding to this drug activator. So that's another example where you can identify a subset of patients, again, just a small percentage of Parkinson's patients, but where we might have greater success in slowing progression. And although some trials have failed, there's a couple others ongoing now, one in particular, also our site's involved in this clinical trial funded by the NIH, of a drug called inosine that increases uric acid level. This is led by Michael Schwarzschild at Massachusetts General Hospital, who's done a lot of the background work along with many others. And this increases uric acid, as I mentioned, which is an antioxidant. And it's thought that oxidative stress may play a role in Parkinson's progression. And there's a lot of data suggesting that low uric acid level may play a role in the progression. No proof in Parkinson's patients, but promising enough that this strategy was tried. But rather than just testing inosine, the drug that increases uric acid level in all Parkinson's patients, for this trial, we first measure uric acid level in a patient who volunteers to participate. And only if their level is low, below average levels, do we enroll them in the study. And that serves two important purposes. One is they're most likely to benefit. They have low uric acid level. They have the most to gain by increasing it. And two is it's safer, because you don't want to take somebody who already has high uric acid level and raise it higher, because that would be unsafe. They'd have a higher risk anyway of gout or kidney stones or other problems. So here's a way where using a biomarker that will allow us to identify patients at greatest chance of benefiting and at greatest risk of tolerating a given treatment. Unlike the genetic causes, this one, although it's only a subset of patients, it's a big subset. About half of all patients would have the potential to benefit from this treatment if it works. But I should mention that I strongly recommend against just going off on your own and trying inosine, because actually it is available, but it would be highly risky for a patient just to go take it on their own from a health food store because of the risk I mentioned, gout, kidney stones, and other risks. And even if you worked with your physician, say, who said, I'll measure uric acid level, I still recommend against that. We have really tight controls within the clinical study to do the best we can to ensure patient safety. But there are still risks, and that's in the face of no proven efficacy. Until we know, some people had recommended CoQ or creatine before we knew. Fortunately, in that case, doesn't do much harm probably either. I think inosine has a little more risk if you just use it, but still with the uncertain efficacy. So I think we need to wait for the phase 3 data before we know whether or not to recommend it for patients.
So it sounds like there's some optimism looking ahead, but don't jump the gun for patients.
That's right. And jumping the gun is an important issue to raise, actually, because a lot of patients, when I tell them, I have a lot of reason for hope. We've got several studies underway, others in the pipelines, but these take years to do. And they say, well, I need something now. And they're willing to take risks. But this strong motivating desire to do something now to slow progression can lead to risky decisions to use unproven therapies that may have risks, sometimes even unknown risks, in the face of unknown efficacy. And I think that's a dangerous path to go down, and some people do that. And I understand the motivation too, but I think you have to be cautious. We don't have what we really want, which is a simple way to take a drug and, in just a few weeks, know if it slows progression. The only way to know is a slow process. But we're getting there. We are making progress. We're much closer than we were before.
Very good. I appreciate it. Thank you.
Research is the only way that new treatments may be found to slow the progression of Parkinson's, and clinical trials are part of that process. If you think you may want to participate in one, call our helpline at 1-800-4PD-INFO to find out about what research is going on in your area. Much of it is conducted at our centers of excellence around the country and internationally. If you want to leave feedback or comments on this podcast or any other subject, you can do it at parkinson.org/feedback. We'll respond to some questions in future episodes. At the Parkinson's Foundation, our mission is to help every person diagnosed with Parkinson's live the best possible life today. To that end, we'll be bringing you a new episode in this podcast series twice a month. In our next podcast, we'll hear about work testing a vaccine against alpha-synuclein. Till then, for more information and resources, visit parkinson.org or call our toll-free helpline at 1-800-4PD-INFO. That's 1-800-473-4636. Thank you for listening.